So my summer project was about developing a pipeline to answer the research question, are HuBMAP donors healthy at the molecular level? A little bit about me, I'm a rising senior at the University of Michigan studying microbiology, Islamic studies, and bioinformatics. This summer, I interned at the Cyber Infrastructure for Network Science Center at the Indiana University Bloomington. Coming into the internship, I knew a couple of skills. I knew basic programming in Python. I knew basic differential gene expression. I also knew basic biology, but I hoped to learn a lot of things. I hope to learn things like single-cell RNA-seq analysis and what software to use. I wanted to learn more advanced Python methods. But most importantly, I wanted to learn more about the HuBMAP data sets, like how to process them, how are they formatted, and how did they come to be from the tissue blocks. I also wanted to learn about the HuBMAP tools, the various HuBMAP tools, and how to use them. So, in essence, the goal of HuBMAP is to create a map of the human body on the single cell level. To reach this goal, we need to get biological data from human tissue samples. So, the primary component of HuBMAP that works on getting biological data from human tissue samples and processing it is the tissue mapping centers at HuBMAP. And I want to emphasize that HuBMAP wants to collect healthy human tissue, and that will be important in a second. How is that healthy human tissue identified? So, a pathologist will review each tissue sample using a histological staining called hematoxylin and eosin staining. The pathologist will look at the staining and identify whether this tissue sample is healthy or not. If it is healthy, the tissue sample is sent to the TMC, and the TMC is a tissue mapping center. The tissue mapping center generates a data set that is submitted to the HuBMAP data portal using the information from the tissue sample. So the tissue donors have various health qualities. They aren't all the same. They're different people, right? So the donors themselves vary in terms of age, sex, BMI, race. They also vary in terms of medical histories, which is things like diseases that they might have had in the course of their lives, and social histories, which could be things like whether they were smokers or drug users in their lifetime. It's important to note that not all donors have all donor metadata info available in the donor lineup that's in the figure on the right. Some of these fields are optional, like cause of death and medical and social histories, which creates a problem when you're trying to accurately look at a donor and identify what type of donor this is, right? If they're really healthy or not. So I want you to notice in the figure on the right, the medical and social histories in the figure one. So, for example, we have some donors over here that have diabetes, one that has coronary artery disease, and another that has gastrointestinal disease. And it's important to note that the samples that came from these donors were marked as healthy by the pathologist, but the donors themselves don't actually seem so healthy. So this is where my research question comes in. Are the HuBMAP donors healthy on the molecular level? We will be comparing gene expression profiles of kidney blocks from donors with various social and medical histories. But since that question is so big, the goal of my internship itself is to just develop a pipeline to try to answer the question that you can apply to different data sets to try to answer the question of, are HuBMAP donors healthy on the molecular level? So we need to break this down into smaller questions to be able to answer this question, to be able to create the pipeline. We need to, for example, learn how to process multiple data sets into human readable format. We need to compare the data sets on some information like tissue block, location and size, cell type populations, and highly variable genes. We also need to be able to compare the donors on some metadata like their medical histories and their age, etc. We need some materials, first of all. We need to download HuBMAP datasets with gene expression data. The HuBMAP datasets must be from the same organ, they must be processed with similar technology, and the sample tissue block must come from a similar location within the organ so that they won't vary too much. We also need donor metadata from the data portal donor lineup in the HuBMAP data portal. So to start off, we chose 6 different HuBMAP datasets from the left kidney to analyze. Uh, as I said, these were all from the left kidney, and they were sequenced using single nuclear RNA sequencing. So how did we initially start processing these HuBMAP datasets? We programmatically processed them by first downloading the processed secondary analysis.h5ad and data files from the data portal. We loaded those datasets into a Python interface. We imported ScanPy, which is the tool in Python that we'll be using to analyze the single cell data. And then for each dataset, we calculated some highly variable genes using a ScanPy function. And now, after all that is done, our data files are pretty much ready, and we can go on a deeper level and start analyzing what they actually look like. So, just for reference, what do the data sets themselves look like in the data portal tool? This is what they look like. This is an example data set in the data portal tool, and this file that I circled here in blue, the secondary analysis file, is the file we chose to use for processing because it is already processed, like it's already done all the pre-processing and quality control, etc. So first of all, we need to compare our data sets to make sure they're as similar as possible, or at least we know the differences if there are any at all, right? Because we're trying to eliminate all those differences between the data sets so we could just focus on the differences between uh, the data sets, gene expression profiles, uh, and if they have a correlation to the differences that are occurring among the different donor metadata. So this table I got from the HuBMAP Entity API. I programmatically got it from the HuBMAP Entity API, and Andy Buchel from the CNS lab over here published a script in GitHub that I modified slightly to output this table for us. It's important to note that these two green rows right here are from the same donor. So now let's compare our donors themselves. So this is just comparing our donor metadata. This is something important that we need to do to make sure we know the differences that are going on between their medical histories and their age, etc. This table itself, the information in this table was collected from the donor lineup in the data portal. And now the next step we need to do is to compare the location from which our tissue blocks came from. So we're looking at which structures the tissue blocks collided with. You can see over here that the first 5 entries, or the first 5 rows, pretty much have a similar, similar structures that the tissue blocks collided with exception to the final row right here. This one did not collide with the renal medulla, in contrast to the other structures that all collided with the renal medulla, while this one did not. So I'm highlighting it because we will look more into it in future slides. So now we're visualizing the tissue block locations actually in 3D using the registration user interface. You can see that the yellow block is the tissue block and where it's located in the kidney. Uh, uh, Panels A, B, and C, pretty much, if you notice, they have a similar location that the tissue blocks came from, whereas D, E, and F are kind of different in where those tissue blocks came from. So we expect their compositions to be kind of more different. C, the panel C that is highlighted in red over here is that same tissue block that did not collide with the renal medulla. So now we're going to take a closer look at the 3 tissue block locations in the exploration user interface. You remember those 3 that I told you about that were very close to each other in location? These are those 3, and we've zoomed in on them to look at them at a more close level. The yellow block over here is that same tissue block that did not collide with the renal medulla. At this point, we want to compare the compositions of the 6 different tissue blocks in terms of their cell compositions. On the figure on the left, the y-axis is the cell count, and the x-axis is the specific data set. For the figure on the right, it's normalized on cell count, so the proportions are out of 100%. The colors are different cell types. I've highlighted in the red box that same tissue block that didn't collide with the renal medulla. So you can see how it differs from the tissue blocks that do collide with the renal medulla. So similar to the previous slide, these figures are showing cell type compositions, but this time we filtered the data sets on the cell types that are common amongst all data sets. I again highlighted that in the red box, that same tissue blocks that didn't collide with the renal medulla. And notice how it looks a lot more different in cell composition than the other 5 data sets. So especially in the figure on the right, you can see how much more different it is than the other data sets in terms of cell composition on the cell types that are common amongst all data sets. So now finally, for our last piece of analysis, we went deeper into the molecular level of those data sets to see how the gene expression profiles for how the gene expression profiles vary for each of these data sets, top 50 variable genes. So on the x-axis, we have the top 50 highly variable genes for that. Sorry. So on the x-axis, we have the top 50 highly variable genes for that data set. And on the y-axis is the cell types that are common across all 6 data sets. This specific dot plot that we're looking at is for that same tissue blocks that didn't collide with the medulla. We're just showing it as an example. We have 5 more of these dot plots. So to actually see if you can see a significant difference in the gene expression profiles between these data sets, you would compare all 6 of these dot plots that we generated. So yeah, we would have to, so we would see if these data sets are any more similar or different, and we would try to correlate similarities or differences with the specific donor metadata that's going on. In conclusion, overall, we worked on discovering a best path to take to build a pipeline to answer the question of, are HuBMAP donors healthy on the molecular level? Something interesting that we found out was that the majority of those top 50 highly variable genes produced in ScanPy for each of these data sets were mostly long non-coding RNAs, pseudogenes, and novel transcripts. These types of transcripts are not highly researched in contrast to those highly researched genes that we always see in literature, and they are always like significant when it comes to various health conditions. But no, those top 50 highly variable genes that we saw in those dot plots weren't at all those highly researched genes that we're used to seeing. But it's important to not ignore these transcripts because they could be important. The pipeline to answer this research question is incomplete, but the goal is exciting. We need to look at more data sets and add more analysis methods to the pipeline to be able to perfect it. Something important that I learned how to use the HuBMAP data and the tools, for example, the HuBMAP data portal, the entity API, the registration user interface, and exploration user interface. And I also learned more bioinformatics tools and methods like how to use ScanPy and Python to implement single-cell RNA-seq analysis. For future work, we want to complete the pipeline tool to make it as accurate as possible. We also want to filter the gene expression profile figures on genes that are known to be important in kidney function. So you remember how I said that right now they're mostly like long non-coding RNAs and pseudogenes, etc. We want to filter them so that the highly variable genes that are showing up in our gene expression profile figures are the genes that are like commonly known, right? And also we want to filter them on the genes that are like correlated to diseases. We also want to find donors that have more metadata because that is more valuable information for us. And we want to find and compare donors with similar medical and social histories. Like, for example, we want to compare a coronary artery disease patient to a coronary artery disease patient so we could see how similar they actually are. Because if they are very similar, then that's a good sign. That means our pipeline is doing what it's supposed to do to identify the differences in gene expression when it comes to donor metadata. And we want to also identify data sets that are more similar in location and size so we could try to eliminate all those differences that come from the data sets themselves. Like I don't want differences showing up because my tissue blocks were from different locations. No, I want the differences to show up because my donors are different. My one of my donors has this medical condition while my other donor is more healthy. Finally, we want to use other data repositories to compare our data sets to, like GTEx and Cell by Gene. For acknowledgements, I want to thank the CNS lab and HuBMAP for accepting me. And also, I want to thank Ellen for being an amazing mentor. And I also want to thank the HuBMAP internship program for the team for being amazing mentors and helping us all navigate our internship. Any questions?